to our next chat, which is by Leah Oppenheimer. She's a summer associate with us here at LDV. She's about to begin her first semester at Columbia Business School for her MBA, and she spent part of her summer really delving deep into how phenotyping and genotyping combined can advance the plant breeding process. So she's going to be sharing some of our findings from our upcoming LDV Insights report on automated phenotyping. Leah, take it away.
Thanks, Abby. So as Abby said, today I'll be talking about how visual data is enhancing phenotyping to help make sure the world stays fed. So you probably all know that Earth's population is growing, but at the same time, Earth is not getting any bigger, and as a result, arable land is decreasing on a per capita basis. So we need to be smarter about how we're growing food in order to keep everybody fed. Now, plant breeding has been around for thousands of years, but a little more recently, it's been unlocked by something called next-generation sequencing that uses fluorescence imaging and allows us to map out the plant genome. Using techniques like CRISPR-Cas9 and genetic modification, breeders can breed plants according to specific traits. So that could be color, it could be taste, it could be yield. Now, plant phenotyping is a really critical part of the plant breeding process, and it refers to the measurement of various plant traits. So we have leaf color, kernel count, stem thickness, and leaf angle. And this is really critical because it allows us to understand how plant genes react with their environment, factors like irrigation, soil, sun, and temperature. The problem is that phenotyping has historically relied on manual measurements. So what you see here is a breeder matching the color of a leaf to a color card in order to understand how much nitrogen to give in a fertilizer application. The people making these measurements are really highly skilled operators. They're breeders, they're scientists, and as a result, having this done manually is really expensive, and it's also time consuming, and it's a bit imprecise. So this problem altogether is known as the phenotyping bottleneck. Now, the good news is that visual technology is really unblocking that bottleneck. So what you see here is a tomato plant imaged using 3 different types of visual technology. On the left, we have near-infrared imaging. Water absorbs near-infrared light, and as a result, this type of imaging tells us which parts of the plant have the highest level of moisture, which you see in those dark spots in the stems and the leaves. The middle image shows the same tomato plant imaged using RGB or visible light imaging, which is the same way that you and I would see the plant, but because the computer is looking at it, it can tell us much more accurately how green those leaves are and how big they are, for example, or maybe how thick the stem is. On the right, we have the same plant again, this time with fluorescence imaging, which shows us how much photosynthesis is happening. When chlorophyll is excited, it emits fluorescence, and by capturing that, we can see in those reddest spots of the leaves where the most photosynthesis is happening. So all of this is to say that visual sensors and computer vision are enabling us to measure plant traits, including traits that we couldn't see as easily before, and it's enabling us to do that more precisely. And we can also correlate them back to genomic data to really capture that picture of how those genes are being expressed in the environment. So on the right, we have a couple of examples of the imaging types used in automated phenotyping. We talked about RGB, near-infrared, and fluorescence imaging with the tomato plant. There's also hyperspectral imaging, which captures light across the spectra, and that's used to measure physiological traits and stress response. There's also thermal infrared imaging, which lets us capture temperature. As we build out these pools of data, we'll better be able to predict what a plant will look like at full maturity, even from a really young age, which is really cool. Now, this technology is already being rolled out across large greenhouses globally, greenhouses being the first places, where the first stage of phenotyping takes place. Smaller greenhouses, which are responsible for more specialized seeds, the rollout there is going to depend a little bit on the cost of technology and the development of the software and pipeline for connecting phenotyping and genotyping data. We also expect to see more business opportunities here in 3D modeling and predictive analytics. After the greenhouse, phenotyping moves to the open field where plants grow in their actual growing environment. This is the second stage. We're already seeing entrepreneurs working on bringing automated phenotyping to the field, And we expect to see other business opportunities across robotics, drones, remote sensing, and edge compute that's really going to enable this space to grow over the next 5 years and beyond. So all of this technology stands to have a huge impact across the global seed market, which is worth $42 billion. So right now it can take up to 10 years to create a new breed, and we expect this to help reduce that time. It's going to improve the quality and quantity of data across phenotyping and genotyping and really allow us to build that correlation between the two. It's going to optimize expenditure and cut costs across nearly $4 billion spent on seed research and development annually. And it's going to reduce the land needed for breeding experimentation through precision data capture. So in conclusion, we're really excited to say that visual data enhanced phenotyping shows immense opportunity for investment. And it's also going to enable us to continue to produce enough food for the whole planet. Thank you.
Thank you, Leah.